Good afternoon, everyone, and welcome to this Thrombosis Week session on thrombosis research. I'm Dan Horner. I'm a consultant intensivist and emergency physician working in Manchester in the UK, and I'm joined by Dr. Lara Roberts, who's a haematology consultant with a specialist interest in thrombosis and hemostasis working in London. I'm going to share my slides and see if we can operate the tech. We've got about half an hour to talk to you about some cutting-edge thrombosis research and to talk about the concept of network research in thrombosis, which has taken great strides recently. Thanks all for joining this session. I'm sure it'll be a good one. Hi everyone. I've been asked to just remind you all that if you have questions, please do pop them in the chat. Joe Jerome is curating brilliantly in the background, and Lara and I will try and pick up your questions in the chat as well and answer them as we go. So first challenge, let's get the slides on. Okay, I'm hoping that you can see my slides there, the first one. So this is going to be just the first 20 minutes really talking about some current ongoing thrombosis research projects and then talking a little bit about things that are about to unfold and start recruiting in the future. And I'm very happy to take questions about any of them or talk about any topics aligned. First trial that I'd like to talk about, which is already recruiting patients and was conceived and funded in October 2023, is the TiLLI trial. So this is a trial looking at thromboprophylaxis for patients placed in lower limb immobilization. And we know that this is a slightly difficult issue as international variation in practice. There's a relatively low VT event rate from placing patients in a lower limb immobilization device like a boot or a plaster cast or a cricket pad splint. But we keep seeing fairly catastrophic consequences of this in the form of patients who aren't really risk assessed for VTE, who aren't told about the potential consequences. And so when they get more pain or swelling to their lower limb in context of injury, they don't tell anyone, and the disease unfortunately progresses to the point where medical services can't do anything about it. And these are just a kind of smattering of recent cases that made the media over a decade, and unfortunately we do continue to see index incidents both in international community and in the UK as well. We've been looking at this issue for the last 15 years or so, I would suggest, and we published multiple evidence reviews and an evidence synthesis funded by the NIHR several years back, looking at the clinical and cost effectiveness of thromboprophylaxis. And we know that thromboprophylaxis probably does reduce the risk of VTE by about half, in the same way that it does if you use it for hospitalized inpatients or other high-risk groups. But the challenge is that the low event rate means that event rate goes from a low number to a lower number, and the costs of medication and the potential risks or side effects need justifying with trial evidence before we can roll out national practice. So we've been funded by the NHR to start the TiLLI trial, which is a multi-center study, and we've designed the trial to split into two different risk strategy groups based in line with current UK practice as advocated by NICE. At the moment, what NICE recommends is that we do a risk assessment on people in lower limb immobilization. And if we deem them to be at high risk, we offer them some form of pharmacological thromboprophylaxis. And here we think the question really is whether we continue to use low molecular weight heparin daily injections, which is the current standard of care recommended by NICE, or whether there's a potential role to use DOAC therapy, so oral tablets, which are cheaper, preferred by patients, and other research areas would suggest they're probably as effective, if not perhaps more effective, than low molecular weight heparin injections. But we don't have any real trial evidence looking at DOACs in this patient cohort, so hence the trial and hence the design. In the low risk group, we're going to approach things slightly differently. So as per UK standard of care, we're going to assume that the current standard of care is conservative management and no pharmacological thromboprophylaxis, lifestyle advice only. But we're going to include randomization arms then to DOAC or low molecular weight heparin to try and test the superiority hypothesis that some form of thromboprophylaxis might benefit those lower risk patients. And we're including that group because we know from previous cost effectiveness work that it might actually just be more cost effective to offer thromboprophylaxis to everyone rather than try to cleverly risk assess and perhaps sometimes get it wrong, sometimes get it right. And we know that different patients have different understanding and feelings about their risk level, and perhaps offering pharmacological thromboprophylaxis to those people who are calculated to say are at low risk but have particular demographic features or other characteristics that might increase their risk may be a plausible option in the future. We've set this up as a low-touch digital trial in the main, which means there's online resources, so a patient video, there's patient information sheets, patient resources mapped to 4 common languages in use in the UK. And all of those resources are free for you to access and look at now under tilli.digitrial.com. And that's essentially a digital version of the patient information sheet, as you might suspect for any study, that captures, I think, the difference in the 2 trials quite well. And there's a really nice 2-minute video explaining the trial, which we use to introduce the concept as we approach patients. And then the light touch continues throughout the study. So after the consent and randomization process, we collect a little bit of baseline data, and then we have 7-day questionnaires that are mostly completed by participants. So our database automatically emails the case report forms and the necessary surveys to the participants, and they can complete online from the comfort of their own home with a nice cup of tea. So helpful research. And we're testing the adherence to therapy there. We're testing weight-bearing status and management, because we know that the decision to immobilize on day 1 sometimes changes at day 7, day 14, day 21. And we're also looking at serial quality of life measures throughout the study as well. And then, of course, at a later stage, 42 and 90 days, we're going to look at VTE and bleeding outcomes in line with ISTH definitions. And how are we getting on? Well, it's all very exciting. TiLLI's been recruiting probably for about 6 months now, since the initial pilot was given green light. And we have 9 active sites that have been set up and have green light from our sponsor, with variable levels of recruitment and some challenges, which I'll come on to. But overall, we are just about meeting our green criteria for progression for TiLLI High. So we've recruited over 120 patients now. We're matching our curve, and all recruitments are picking up with increasing experience. And we're finding that the conversion rate to approach and recruitment is very high in the high-risk group, because patients are naturally interested in the idea of taking a tablet when the hospital would usually offer injections, and are often happy to participate in research. TiLLI Low has been trickier, and our numbers are lower than expected there. And we think that's a combination of a number of things, really. So when we explain to patients that they have a relatively low risk of VTE in line with these kind of findings here, often when you approach them to participate in a randomized controlled trial that might involve then using a drug for several weeks, the initial response is to be relatively reluctant and to take the small percentage risk here at face value and seek help and care and attention as needed. And of course, that's general practice in the UK, so that's a perfectly reasonable approach to this. Some of this, however, is involved with the setup and design of the trial. So we found that most of the study is being recruited in minor injuries areas within emergency departments nationally. And that injury units are staffed predominantly by emergency nurse and advanced practitioners, many of whom are unfamiliar with trial recruitment in patients on drug trials. So we're taking steps to train up more people and allow more leniency in who can approach and confirm eligibility and who can prescribe these trial medications. We've been working really closely with our patient advisory group to get advice from them about the trial design of TiLLI Low and what kind of things we think might be helpful in terms of increasing our recruitment rates. And we've held multiple investigator meetings across the recruiting sites to talk to them about their strategies for recruitment and see how we can optimize them. We need more sites, we need more patients. And people are very welcome to get involved. So the aim for TiLLI is to recruit across 30 NHS trusts. We've got a long list of potential sites to be involved that we're working through, and I think we've held site initiation visits now for well over 15, but there's certainly room for more. So if anyone would like to know more about the TiLLI trial and get involved, you're very welcome to contact me direct. But the TiLLI managers are likely to be more responsive and perhaps even more helpful for logistical queries about how to get involved, and they can be found at the middle email address right there on your screen. Got 10 minutes to talk to you about 2 other trials. And the second one is a funded trial that's about to start. It's a bit of a gear shift away from a lower risk population, the emergency department, and a plaster cast. And here we're talking about patients with a traumatic brain injury. So those patients who have sustained a significant head injury and are admitted to a neurosciences hospital, usually via a major trauma pathway or a self-presentation and subsequent secondary triage. And we know that when people have a bleed on the brain, that there is a risk that bleed could get worse. And we want to take steps to mitigate and control that and ensure the best possible outcome from a TBI perspective. But we also know that this population is very high risk for VTE. We've quoted event rates of around 10% at 90 days in the literature previously. And we're seeing emerging evidence that thromboprophylaxis may stand to benefit this population. Many of you, I'm sure, will have worked in neuroscience and trauma centres, and you'll be familiar with the daily dialogue of when it's safe to start pharmacological thromboprophylaxis in the major trauma patient, how we balance the risk of bleeding versus the risk of clotting, how a lot of subjective opinion can feed into that and individual concern. And actually that translates to a relative lack of consistency in our NHS practice. And this is a recent systematic review and meta-analysis that just perhaps captures the slight change in thinking over the last few years. So you can see from the top forest plot here that the risk of hemorrhagic progression in TBI actually isn't particularly any higher with, sorry, the bottom forest plot is hemorrhagic progression. You can see that that's not particularly higher with early or late commencement of thromboprophylaxis when we're talking about 72 hours or less than 72 hours as early. But you can see from the forest plot at the top that the VTE risk actually significantly reduces, halves the relative risk ratio there at 0.38 when prophylaxis is started within 72 hours of the TBI, rather than deferred to 5 days or even not given at all, as often happens in clinical practice. So this is a real conundrum for clinicians and a real issue for patients. So I've supported a group of neurosurgeons from Cambridge who've led the TOP TBI study to develop a protocol here, succeed in funding application and succeed in trial design. And the website for TOP TBI is listed here at the bottom right-hand corner. You can take a look at that. The protocol is published there, which is a lengthy document. But in brief, what we're looking to do is take patients admitted with traumatic brain injury to any of the major trauma neuroscience hospitals in the UK. And we're going to randomize them to start their thromboprophylaxis at either a point less than 72 hours from injury or a point of 5 days or more, or by clinical discretion. And we had lots of discussion about the separation points for those interventions. We wanted to assure ourselves that there was time for the early prophylaxis group to have serial CT imaging to confirm stability of the intracranial hemorrhage. And we wanted to assure that there was a period of predicted clinical interventions such as ICP bulb placement or even external ventricular drains. And then for the delayed phase of care, we wanted to assure ourselves that this would be a separate intervention, because of course we can't design a randomized controlled trial to start thromboprophylaxis at 71 or 73 hours. So there has to be separation of the intervention to assure that we're actually testing a valid hypothesis. Population are going to be those with TBI who require admission to neuroscience centre. The intervention and control group are as defined there, and the outcome, as you would expect, is clinically relevant VTE within 30 days from randomisation was the NIHR advice here. And we're going to also follow up, of course, for hemorrhagic progression via CT and all clinically relevant endpoints, including mortality. There's going to be a reasonable level of equipoise in this group and challenge, because we know that some neurosurgeons have very strong feelings about the risk of bleeding, and others, perhaps intensivists like myself, have very strong feelings about the risk of clotting. And we need to assure ourselves that we're striking the right balance in terms of meeting that equipoise. So within the exclusion criteria, we've actually added additional levels to clarify that if the treating clinical team lack equipoise on either of those facets of care, that we shouldn't necessarily recruit these patients. So we're really looking at that group where there's clinical uncertainty about the right thing to do. And at that point, we would always, because clinicians in my unit say, rather than provide random care, we should randomise, and the TOP TBI trial gives us an opportunity to do so. And in terms of addressing equipoise, we work very hard with the relevant stakeholders to try and obtain support and disseminate this for our stakeholder group. So these are just examples of supporting letters for the original funding bid from the Society of British Neurological Surgeons, from the Intensive Care Society, and Thrombosis UK. And this support's been hugely helpful in gathering momentum, launching sites, and getting up and running. Our website has been designed, our protocol has been written, our database has been built, our CRF is designed, and Cambridge as the leading site are looking to open towards the end of this month. So this should be hitting the shop floor within the next 4 weeks or so, and we're all excited to get going with it. And then the last few minutes is about something that's been recently funded, but the protocol hasn't started drafting yet, so this is very much still in the inkwell. But we know from recent advances in VT diagnosis that there are lots of different ways to do it, I suppose is the short-term message for emergency physicians. We know that we've seen lots of evidence recently about the importance of assessing pre-test probabilities in the Wells score or Geneva score, and then basing further investigation for suspected PE or DVT then on the D-dimer level in those patients at low to moderate risk, or based on definitive investigation in those patients who are deemed to be at higher risk. But interestingly, over the last 5 to 10 years, we've learned that the D-dimer is not a dichotomous variable, it's a continuous variable, and rather than it being positive or negative, we can perhaps adapt it to either age thresholds or clinical probability thresholds, or even perhaps clinical gestalt, to use it in a wiser fashion and to reduce the number of patients who are referred for definitive imaging and have to wait or be exposed to the harms of radiation and contrast and cannulation and long stays in hospital. And we also know that with waits for duplex ultrasound and CT imaging, we could significantly reduce the amount of interim anticoagulation provided to this group if we can find a way to rule out PE and DVT that refers less people for scans. And when this has been studied in contemporary work published in high impact journals, what you find is that a D-dimer strategy adjusted to clinical pretest probability might save chest imaging to the tune of several hundred patients in a group of a couple of thousand. Or if you adjust the D-dimer level to an age-adjusted threshold, as recently supported by NICE guidance, you can significantly reduce the number of onward referrals there. And there are different clinical risk scores, like the years score, or like the Geneva score, and like the adjust PE score, which just takes a particular facet of the original Wells and looks to use that to classify people into high and low risk groups. And all of these strategies stand in observational research to reduce the amount of patients referred for imaging in a relatively safe manner. When we looked at systematic review data and meta-analyses on this work, or when we've pulled the observational evidence together, what becomes clear is that although you increase the efficiency of your test strategy by raising the threshold for D-dimers being positive, that sometimes carries an additional failure rate. And how high that failure rate is in terms of how acceptable that would be to clinicians and patients is very much up for debate. Because some people have very strong feelings about the need for definitive investigation, and some people are very keen to avoid waiting in hospitals or having injections to thin their blood if their risk of VT is actually quite low, or if the risk of failure of that diagnostic strategy is very low. So we need more research to find out. And we've recently been successful in applying to the Royal College of Emergency Medicine for a grant to assess just that. And we're very pleased with ourselves for thinking of the acronym DAVE, which is for Diagnostic Algorithms for VT in Emergency Care. And our proposal is to perform a large-scale multinational observational cohort study where we capture upfront data on clinical risk prediction variables. We look at the D-dimers that are sent on patients attending emergency departments. We correlate that with the gold standard investigation, so we're not going to change anything regarding clinical practice in the study. We're just going to allow it to unfold as it usually would. But then after we've collected a large sample, we can report back on the efficiency and the failure rate and the tolerability of various diagnostic strategies, including age-adjusted D-dimers, clinical probability adjusted D-dimer thresholds, adjust PE, so gestalt influenced, and the years criteria in a UK population as well. And we're going to need lots of people from that kind of observational research, and we're going to need lots of researchers. And we have delivered this kind of research before in the context of subarachnoid hemorrhage, or in the form of crowding via trainee emergency research networks, or flash mob research, which is increasingly gaining popularity in acute care. And this is the idea that we would engage trainees up and down the country to deliver a high impact study based on a condensed period of data collection of only maybe 3 months or even as less as 6 weeks, depending on the numbers. Because we know that most emergency departments will see 10 to 15 patients per day at least, presenting with suspected DVT or PE. If we can recruit across 100 departments, and we can recruit across 100 days, then you can do the math on that. It's over 10,000 patients, which gives us quite a lot of power in our research findings, and it helps ensure that any conclusions we reach are relatively robust. We've been working with HemeStar, which is the hematology equivalent trainee network, to develop our protocol and proposal, and they're named and listed on the original funding application. And we're really looking forward to bringing these two networks together to try and deliver more information on this important topic. Contact details for their networks are below, but TReN will be driving the protocol development and inquiring about sites. So if anybody wants to get in touch and express interest, I suggest you email the TReN email address, which is just listed at the bottom of the screen there. They're the only slides I had, and I will stop there to give Dr. Roberts now the last 5 to 10 minutes to talk a little bit more about network research and to help create the questions. Thank you very much for your attention.
Thank you. Just bear with me while I share my slides. Okay. Yeah, so I'm just going to spend a few minutes talking about the UK Thrombosis Research Network. This is an informal network that's set up relatively recently, sort of late 2022, 2023. And it was set up by a group of us who were then involved with the NIHR Clinical Research Network. Because as you'll all know here, venous thrombosis is highly prevalent. So it's a lifetime prevalence of 1 in 12 adults. And it's associated with significant morbidity and mortality. But when we looked at the hematology research portfolio, it was significantly underrepresented. It's that very tiny slice at the top of the pie chart, with far more research being performed in much rarer conditions. And as Prof Horner has highlighted, VTE is not just managed by haematologists. There's plenty of other interested clinicians involved in thrombosis research. And that might be one of the contributors, although this is supposed to incorporate studies that were supported by haematology rather than just led by it. So we set up a steering committee back in early 23 with representation across the UK, including some trainees. We've got a non-haematologist, a basic scientist, and Joe representing the patient and public voice. And we do have fairly broad membership, and I'm really pleased that we've got over 1/3 of our members are non-medics. And we have representatives from other specialties outside of haematology who are also interested in thrombosis. And the aims when we set this up was to create a multidisciplinary network of people interested in thrombosis, so not just being haematology focused, and also to join Invent VTE, which is an international network of VTE research networks, to increase or enable participation in global non-commercial studies. And the longer term, we'd like to obviously increase the number of RDN portfolio studies in thrombosis. And it's great to hear the studies that Prof Horner is involved with, because I think just those studies alone more than double the current number of studies on the portfolio, and also to strengthen collaboration between clinicians and scientists. I just want to briefly talk about Invent VTE. I'm not going to read through all of their aims. These are available on their website if you want to have a closer look. But they do really encourage participation of early career researchers. They have a number of members across Europe, North America, and including Australia and New Zealand and China. The UK is the newest member. And there are some funding and career development opportunities available. If you're going to ISTH, I highly recommend going to the Dragon's Den session. This is a really fun session where early career researchers will pitch a proposal, which the audience then votes on. And the winner receives some startup funding to develop a pilot project based on their proposal. And they have twice yearly Kickstarter awards, again, to set up pilot studies across an international collaboration, so involving at least 2 members of the network. And then Murray Thomas has one of these awards with Canada, looking at anticoagulation for cancer-associated thrombosis in patients with thrombocytopenia. And they also fund training fellowships. There are lots of examples of successful studies that have been run with InventVTE support, so particularly in COVID, but also the HILO study that many of you will be aware of, looking at dosing of low molecular weight heparin in pregnancy, and an observational study looking at DOACs in splanchnic vein thrombosis as examples. The SAVOR trial is an ongoing randomized controlled study looking at rosuvastatin versus placebo for prevention of recurrent VTE and other arterial events following a primary VTE. This has been run by Canada, and we've recently been awarded funding by the British Heart Foundation to bring this study to the UK. It's just going through contracting now, but we are hoping to open 6 sites in the UK later this year. And we're also partnering with BT Dublin, who have run an educational meeting for the last 10 years in Dublin. And this year we're bringing it to London. We've got some really great international expert speakers on thrombosis joining for that. So I'd encourage you again to sign up if you're interested in attending. It'll be a 1.5-day meeting, and the QR code there goes to the booking link. So if you are interested in thrombosis research, we'd love to have you join. If you're based in the UK, you don't need to have any particular qualifications just to be interested in thrombosis research. It is an informal network, but we do send out emails about people who are looking for sites for studies, and also communicating about Invent VTE opportunities. I'm going to stop there so that we've got a few minutes to answer some of the questions, which I think are mostly for you, Dan. So I'll just go through them. I think we've probably got a couple of minutes, but there's one about the TiLLI trial, commenting that the protocol's really recently changed to reduce the TripCast score from 7 to 6, and just asking if you can explain why that decision was made.
Yeah, it's a good question, and one that has been talked about. I suppose I'd start by saying the protocol's never changed. It's always been 6. The question has arisen because the TripCast score, which is the risk assessment tool we've chosen to discriminate high and low, has recently been published on MD Calc, and the authors recommend a threshold of 7 or above as high risk. Now, any score it is not dichotomous. It is not yes or no risk. It's a numerical score usually, or an ordinal score. And at some point, researchers have to pick a point where they think that threshold is above the risk threshold that's tolerable, and then they say the score is positive. But we actually looked at the TripCast by number and the event rates associated, and we found that with a score of 6 or above, that met our previous estimates for the best clinical and cost effectiveness for use of thromboprophylaxis. So we've detailed this within the protocol for TiLLI, but we think 6 and above probably represents a better trade-off for patients in terms of the risks and benefits of anticoagulation. So that's where we're starting on. And we are increasingly needing to make that clear to sites, because when they go to MD Calc, it gives them slightly different information. We've got some plans to write to MD Calc and talk to them about it. And Delphine Doulier, who is one of the original authors of the TripCast-GAR, is part of our steering group for TiLLI, and has been helping us with that as well. So it is something we've considered. I hope that answers your question. It's a good question.
Thank you. We might just squeeze in one more. I don't know if you want to redirect this, but someone's asking, does it matter which reagent and analyzer for the D-dimer that you use? So they haven't adjusted for age in their centre because they don't think their assay has been evaluated in trials.
So well, I think it's a good question, isn't it? And Lara, I think either of us could say that there probably will be a British Society of Haematology guideline on D-dimers coming quite shortly that we've both been involved in, which talks a little bit about harmonisation and standardisation and the challenges of using different assays. But the question I think represents what's happening in the real world is that while we've got this guidance on how to age adjust, how to adjust to clinical probability, how to adjust to Gestalt, everyone uses slightly different assays for their D-dimer, and lots of trusts don't have the confidence in that research or the nice recommendations even about age adjusting with their particular assay to introduce that change in practice. So I'm hoping that Dave will capture a lot of that. So it will look at what people are doing nationally, and it will record the outcomes and the D-dimer levels and the gold standard investigation for all these patients. And then we'll be able to report back at a trust level to say, had you age adjusted, this is what would happen to your patient cohort. You know, you would have been able to avoid this many CTPAs, or it might say you could have avoided this many CTPAs, but you would have missed these two PEs, you know, and that's really valuable information, isn't it? I think sometimes in the research world, we come up with different ways of doing things or different risk assessment strategies, but we don't necessarily test them through implementation research in a wide population to make sure that they're valid and generalizable. And that's what Dave seeks to do, and I think that's what we should do more of with diagnostic research, personally.
Thank you. And so I think we might need to draw the session to a close because there will be another session following soon. But Dan and I are both happy to be contacted directly by email, or you can send questions to Jo and she'll redirect them if we've missed anything that you wanted to raise. And I'd like to thank you all again for joining, and enjoy the rest of National Thrombosis Week.